Welcome everyone. Good afternoon and welcome to the EFSA ECHA webinar on the use of the XETA, the Xenopus Eleutheroembryonic Thyroid Assay, in the assessment strategy of the EFSA Guidance on the hazard identification of endocrine disruptor. I am Maria Arena. I am a Senior Scientific Officer in the ecotoxicology team of the EFSA Pesticide Peer Review Unit. I was involved in the drafting of the ED guidance, and I'm currently performing ED assessment of pesticides for non-target organisms. Today, as you can see from the presentation, I'm not alone, and I'm very pleased to give the floor to my ECHA colleague Sander for introducing himself.
So good afternoon, and thank you, Maria. Good afternoon, everybody, also from my side. So my name is Sander van der Linden, and I'm a scientific officer at the European Chemicals Agency. And also like Maria, I was involved in the drafting of the ED guidance document. And at ECHA, I work at the Biocidal Active Substances Unit, where I'm also involved in the assessment of endocrine disruptor properties of both the active substances as well as the biocidal products. And now I'd like to give the floor to our last colleague joining us here today for this webinar, which is Odd Kienzler. Please, Odd.
Hi, everyone. And welcome. So I am Odd Kienzler. I'm working in EFSA as a scientific officer in the pesticide peer review unit in the ecotoxicology team. And I mainly deal with the assessment of endocrine disrupting properties of pesticide for non-target organism. And today I will be the Q&A moderator of this webinar. And first I will start by giving you some technical information. So please note that this webinar is being recorded and it will be published afterward on the EFSA website. Be aware also that we are currently working from home, so we apologize in advance for any issue that we might have due to this particular situation. Regarding the question you may have, you are obviously welcome to ask them in English in the Q&A chat. And you can use this chat for both questions on the webinar content or on any technical issue that you might have. And in this case, our support team will deal with those questions and help you. For the question on the webinar content, I will either answer them directly in the chat or we will keep them to be addressed by Maria and Sander after their presentation. So don't worry if they are not answered immediately. We will try to answer all your questions today. If we don't manage because we are running out of time, we still have the possibility to answer them later by mail, as long as you are not connected as anonymous, because in this case, we cannot identify who is actually asking the question. I think that's all what I wanted to tell you, so we can now move on with the presentation. So please, Maria, the floor is yours.
Thanks, Odd. So in this first slide, you see the agenda of the webinar. So I will give you a little bit of background, then we will discuss, I will present the principles and limitation of the XETA. Then I will go through the first case where we think the XETA is a suitable test and can be included in the assessment strategy of the EFSA guidance. At this stage, I will give then the floor to Sander, who will talk, discuss and present the second case for the consideration, the use of the XETA, and then we will have a Q&A session and conclusion. So in 2019, and you may all know this, OECD published the new test method on the Xenopus Eleutheroembryonic Thyroid Assay. From this moment on, I will just use the term embryo instead of eleutheroembryo, since it's easy to pronounce. But you know that when I talk about embryo, I refer to eleutheroembryo. So the publication of the test guideline and discussion had with member states and stakeholders triggered the need for clarifying how to consider the XETA in the assessment strategy of the ED guidance. And to this aim, EFSA, in consultation with ECHA, drafted an annex to the ED guidance. This annex underwent a targeted consultation with member states and the ECHA ED expert group and Environment Working Group of the Biocidal Product Committee. The webinar of today aims at informing you as stakeholders on how to consider the XETA in the ED assessment. So when really the approach that we recommend for considering the XETA as a suitable test. Following the webinar, the annex will be finalized by considering all the feedback that we have received so far, and finally it will be published. So let's discuss a little bit on the principle of the XETA. So as you can see from this slide, the XETA uses embryos from stage 45 to stage 48. These embryos are not able to feed independently, but they still use the vitellus reserves. And this is the reason why they are considered as, the XETA is considered as a valid alternative to vertebrate testing, simply because according to the definition given in the directive, in the EU Directive 63 of 2010, when embryos are not able to feed independently, they are not considered animal. So this is quite an important aspect of the XETA. In the slide and in the graph, you also see the entire life stages of an amphibian, including the life stages that are tested in an amphibian metamorphosis assay. The XETA is a 72-hour in vivo aquatic screening assay. And being a screening assay, it is placed at the level 3 of the OECD conceptual framework. This means that similarly to an amphibian metamorphosis assay, it gives information on the endocrine activity, but being a screening assay, it cannot be used to identify an adversity. So it can only be used for the endocrine activity. So the test uses metabolic competent Xenopus Eleutheroembryonic embryos. Being a transcription-based assay, it uses a transgenic Xenopus Eleutheroembryonic line harboring the THB-ZIP promoter, the THB-ZIP GFP genetic construct. This genetic construct is made up of two parts. We have the THB-ZIP promoter coupled to a reporter gene for the green fluorescent protein. The THB-ZIP gene codes for a transcription factor which is associated with amphibian metamorphosis. The amphibian metamorphosis, we all know, is controlled by the thyroid hormone. It is important to note that the XETA is able to measure the capacity of a chemical to inhibit or activate the transcription of the genetic construct, and therefore whether a chemical is inhibiting or activating the thyroid pathway signaling. However, the XETA is not able to distinguish between different modes of action. The endpoint measured in the XETA is the induction of the fluorescence in the embryos. When transcription of the genetic construct is activated or inhibited following the exposure to, for example, a pesticide or a biocide, then the embryos express more or less GFP, and this means that they emit more or less fluorescence when compared to a control. Thanks to the quite robust validation and what is stated clearly in the OECD guideline, we know that the XETA is not able to cover all known thyroidal mode of action. And we know that the XETA is able to cover modulator of the thyroid hormone clearance hepatic metabolisms, modulator of the thyroid hormone metabolisms, including inhibitors of the iodinase, thyroid receptor agonist, and thyroid receptor antagonist. For each of those mode of actions, one or more chemicals were tested during the validation. Similarly, we know that the XETA is not able to cover a mode of action that are linked to that may lead to the interference with the synthesis of thyroid hormones, simply because the embryos that are used, so embryos at stage 45, do not have the thyroid gland fully formed. So the gland is in the process of development, is under formation, under development. And this means that the embryos at stage 45, from stage 45 to 48, do not fully synthesize their own thyroid hormones. So to a certain extent, they may synthesize some thyroid hormone, but they may not be fully able to synthesize their own thyroid hormones. And this is the reason why substance that may interact with the thyroid hormone synthesis, like inhibitor of the sodium iodide symporter or inhibitor of the thyroid peroxidase, are not considered covered by the XETA. Similarly, the OECD guidelines state that the XETA can potentially cover substances interfering with the thyroid hormone transport. However, since no chemicals with those mode of actions were tested during the validation, for the time being and until further information will become available, those mode of action are considered not captured by a test according to the XETA. Again, this slide shows the thyroid signaling, the thyroid pathway cascade. And again, the slide is to reiterate and to put the emphasis on the mode of action that are covered and the mode of action that are not covered by XETA. So the mode of action that are upstream in the thyroid pathway cascade, meaning the thyroid hormone synthesis and the thyroid hormone transports, are not covered by the XETA, are not considered to be captured by XETA, while the mode of action that are downstream in the cascade, so step from 6 to 8, are considered to be covered. Here there is something to say, because although substances like PTU, who is known to be a TPO inhibitor, was tested during the validation and was found positive, we think that at this stage is not possible to extrapolate the result of PTU to all possible TPO inhibitors, basically for a couple of reasons. The first one is that PTU was tested, was found positive in the XETA, but at quite a high concentration. And this could corroborate what we have just said before, so that the embryos at stage 45 to 48 may not fully be able to synthesize their own thyroid hormone. Additionally, we have also to consider that PTU is quite a special case because it is known to act through several thyroidal mode of action, because it is known also to act as an inhibitor of the iodinase 1, and this could be the reason why it was found positive in the validation phase. So the ECHA, now we really enter in discussing how to use the XETA in the assessment strategy of the ED guidance. So the EFSA guidance recommends, and this to first conclude on the ED properties for humans and in parallel using the same data package on mammals as non-target organisms. This recommendation was given in order to minimize the use of vertebrate testing. Because when we are able to conclude that a substance meets the ED criteria for humans and mammals based on the mammalian data package, the assessment does not need to proceed and additional data are not needed to be generated. Only if the criteria are not met for mammals as non-target organisms, then the assessment has to proceed by considering non-mammalian species. Since here we are discussing about the thyroid, of course, amphibians are considered currently the model species. As you can see from the flow chart and the figure 1 of the ED guidance, When the T-mediated parameters are not sufficiently investigated, the guidance recommends to complement the dataset by investigating the endocrine activity, by fully investigating the endocrine activity. I think here it is crucial to say that we are not discussing in general how to consider the XETA in the assessment strategy, because you know that according to the criteria and the guidance, we should consider all the available information, including the XETA when available. Here we are really discussing when the XETA is considered suitable to sufficiently investigate the endocrine activity, meaning that when we have XETA, it may be used as the AMA is recommended at this stage in the guidance, and the endocrine activity is sufficiently investigated. I think this is quite an important point of the discussion today. So the EFSA guidance recommends to proceed in the assessment by generating other data, additional data on non-mammalian species, so on non-target organisms other than mammals, in two cases. The first case is when the ED criteria are not met for humans and wild mammals. The second case will be discussed later by Sander. So in the annex, we included a flowchart. This is just part of the flowchart we split for a better visualization. So the starting point for the case 1 is to check whether the data set is sufficient for the T modality for mammals. So the first point is to see whether T-mediated adversity was sufficiently investigated according to the definition given in the guidance. When I must say that normally, for pesticides at least, the dataset for the T modality is complete, because normally we have a number of studies with different mammalian species and of different duration where T-mediated parameters are investigated. When no effects are identified in any of the available study and any of the available measured parameters, then the ED criteria are not considered met for humans and mammals as non-target organisms. In this case, the XETA is considered a suitable test to proceed in the assessment. Basically, this is supported, this recommendation is, we think, is supported by the high conservation of the endocrine system across vertebrates and by analyses that are available in the literature. However, we also know from the available analysis that there are some cases, few cases, but still there are some, where a different response between mammals and amphibians is expected. And we think that a test according to the XETA is able to cover, to capture those different responses. Basically because we think that a substance acting as TPO inhibitor or NIS inhibitor will be captured in the mammalian studies. Because, for example, we know that rat is quite sensitive on interference with the thyroid hormone production. If changes in T-mediated parameters are observed, but a thyroid mode of action is excluded, then we still think that the XETA is a suitable test to follow up. Just to give you an example on what could be this situation. So imagine that we see a change in the thyroid histopathology in a rat study or in more than one rat study. But the effects are observed at doses that are considered to exceed the maximum tolerated dose. In this case, the thyroidal mode of action is excluded because the changes that are observed in the thyroid histopathology are simply considered secondary to systemic toxicity. So in this case, we think that XETA is may still be considered a suitable test. Of course, when deciding on which test should be performed on non-mammalian species as a level 3 test, it's important to consider all the available information, meaning also information that may come from level 1 data. When I refer to level 1 data, I mainly refer to literature data and ToxCast data, because although we know that there are no standard thyroid in vitro assay, there are non-standard protocols available in the literature, or we know that the ToxCast include a screening assay for the T modality, and those may be used to take an informative decision on the type of on the most suitable test for non-mammalian species, and in reducing the uncertainty we may have. So basically, just to conclude on the case 1, we can say that XETA is considered a suitable test when no adversity was observed in mammals based on a complete data set. Although some effects, I would say some changes in T-mediated parameters were observed, a T-mediated mode of action was excluded. Again, as I said, it is important when deciding on XETA versus an amphibian metamorphosis assay to consider all the available information that may suggest a possible molecular initiating agent. So this was the last slide for the case 1, and I give the floor to Sander for presenting the other case.
So thank you, Maria. So next to the case just described by Maria, we might theoretically also have a situation actually where there is evidence for the thyroid disruption and that the substance is considered an endocrine disruptor for humans, but not for mammals as non-target organisms. And this would be because, for example, the effect would actually occur in mammals because the ED assessment for humans is most likely be based on mammalian studies. But the effect itself is not considered population relevant. And of course, if it's not considered population relevant, then it's not considered at first, and therefore the ED criteria actually not met for mammals as non-target organisms. We acknowledge upfront that maybe case 2 is a bit more of a theoretical case, because we know that the thyroid pathway is actually relatively well conserved throughout the taxa. However, keep in mind that we still don't know exactly what is the impact of thyroid disrupting effects during development and what is the consequence of the health of natural populations. But the adverse effect on reproductive success in natural populations is certainly very plausible. So since this case might occur in practice, we still try to accommodate it into the scheme. Next slide, please. So here you see part of that scheme. Again, we've taken only part of it for visual purposes. It's better, clearer this way, and it's part of the flow chart that we have drafted in the annex, but now for case 2. So as explained, what you see at the top is that in this case, the substance is actually considered an ED for humans, most likely based on mammalian data, but the effect is not considered population relevant, and therefore the substance is not considered an endocrine disruptor for mammals as non-target organisms. And to explain this more clearly, we also put a footnote number 4 into the scheme where we go a bit more in depth in this. But in the case of a negative conclusion on population relevant for the effect for mammals, for the time being, we need to consider this as a data gap for non-mammals, and therefore it will require further testing. It might, for example, be that actually the substance is not an ED for mammals, but can be an ED for non-mammals and with still population relevant effects. So the remaining question that we still need to answer is, should the substance be considered an endocrine disruptor for non-mammals as non-target organisms, which brings us back again to the 2 pillars of the ED criteria, which is both the endocrine activity and adversity. So we need to consider, is there any evidence for endocrine activity in non-mammalian species, and actually does the substance lead potentially to adversity, so population relevant effects in non-mammalian species? And for the ED criteria, of course, we need to answer both. And like Maria explained, the ED guidance in general recommends, in order to save unnecessary animal tests, it's always wise to start with investigating endocrine activity first, unless, of course, if you have already information available for the adversity. And to start with the endocrine activity, ideally, of course, always we recommend going with the in vitro test, so conceptual framework level 2. But in this case, there are no validated OECD test guidelines available for thyroid disruption. And therefore, we go to the next step, which is level 3, and there we have the XETA and the AMA. So we need to decide in which case we can actually use the AMA or the XETA, since these are the only validated test guidelines. Keep in mind again that the scope of this annex is actually about when to choose the XETA over the AMA, which would be the case where we have an effect on that we know based on the other data that it's not acting on thyroid hormone production, because explained the XETA is not fully covering this, and also not on the thyroid hormone transporters. If we can exclude the effect that the XETA not covering, then we might use the XETA to determine whether the effect that we have seen in mammals does also occur in amphibians as non-target organisms. Next slide, please. So that would be the case here. We have ED, T-mediated adversity identified in mammals and considered to be a T mode of action, but it's not relevant for wild mammal populations. So we need to investigate further on the non-target organisms. And again, to be clear, this might be quite an unlikely scenario. If there is also already strong evidence of the thyroid activity, the remaining question might actually be, is there enough evidence for adversity? And of course, in those situations, a LACDA or even an AMA might be a more useful study to conduct. But again, keep in mind that this annex is about when can we use the XETA instead of the AMA. It's not about a thyroid testing strategy in general. To avoid any confusion also on this, we added that clearly to the annex that it clarifies under which circumstances the XETA might be considered an alternative to the test, OECD test guideline 231, which is the amphibian metamorphosis assay. Keep in mind that to keep the decision scheme simple, it's quite straightforward, but always you need to consider all the data in context, or all the data together. So there actually might situations that based on the totality of the data, you might conclude slightly differently. And the same would hold true for the AMA. If you do the AMA, of course, again, you need to consider all the data together. Next slide, please. So as depicted in the OECD test guideline for the XETA, and as explained by Marie in the beginning, based on a principle of the XETA test, the XETA might not be a suitable alternative if the substance is known to act via TPO inhibition or NIS inhibition, or act directly on the thyroid hormone transporters. If that is known, then you might use the XETA to investigate non-mammalian, non-target organisms for your specific substance. And if the XETA is negative, of course, you still need to consider all the data together, but it can be a good indication that actually the thyroid disrupting mechanism that led you to conclude that it is an ED for human health might not be a relevant mode of action for amphibians, and therefore might not be an ED in this species. Of course, there was always still the remaining uncertainty of what about other species, but we have to stick to the models that we have and that we have validated, and that is just a limitation of what we have to work with. Of course, if the XETA is positive, this might need to be followed up with higher TIS studies, so the AMA or the LACDA, to assess the adversity, as the XETA, of course, does not address that question, where the AMA might. Next slide, please. And so this is the context to which we drafted the annex. In the annex, we try to clarify the situations where you might choose the XETA over the AMA, again, to minimize animal testing. Though we acknowledge upfront that case 1 is probably more likely than case 2. However, in both cases, both case 1 and 2, if the XETA is negative, you might have sufficient information to conclude that the ED criteria are actually not met for non-target organisms, not for non-mammals, but remember we started the decision tree with already concluding it's also not an ED for mammals. However, keep in mind that a negative XETA might not always be sufficient. You always need to conclude based on the totality of the data. And therefore, if it's positive, we probably need to produce additional data to investigate the adversity to be able to enter the full mode of action for your specific substance. Next slide, please. So that concludes our presentation for the foreseen annex. So we kindly ask you to let us know whether you agree with the proposal or whether you have any comments. Of course, we're happy to try to answer any remaining questions that you might actually have. Although, as mentioned in the beginning, it might that we can actually not answer all the questions right now, but keep in mind that we'll take all the comments and questions on board when we try to redraft and finalize this annex. And with that, I'd like to give the floor back to Odd, who will take us through the question and answer session. Back to Odd.
Thank you, Sander. So we do have a few questions, 4 questions so far. So the first one is from Thomas, who is asking to you, Sander, if this test can be used to avoid measurement of the T4 and TSH, for example, in the OECD 422 test. So, for example, if we can do first XETA and then an OECD 422 with or without T4 measurement. So what would you answer to this?
My answer first of all would be that probably this is a bit out of scope, because we really start discussing here where the XETA can be used instead of the AMA. That being said, I think in all cases, both AMA and XETA results could feed back into the mammalian set. But I think a negative XETA in itself would probably not be informative enough to start answering that question. Keep in mind, though, that when we talk about the mammalian data set, that usually you don't have T3, T4 measurements present. But in the guidance, we took the pragmatic decision that this is something we can live with, this additional uncertainty, because it's a lacking parameter in so many of the results that we have, but it's a pragmatic decision. But coming back to the question, the scope is really when can we use the XETA instead of the AMA, and I think it's out of scope of the discussion when they can use the XETA to avoid having to measure T3 or T4 in a mammalian data set.
Okay, thank you, Sander. The next question is from Leonard Welch, and it's about slide 11. So Maria, maybe you can answer to that and come back to the slide 11 of the presentation. And he's asking if the arrow from the additional mammalian data generation should go to the second diamond and not to the AMA.
Well, I think it is not very clear to me. So the arrow from here, I think from
the additional mammalian data generation. So additional data need to be generated for mammal.
No, because here in this case we don't generate additional data for mammals. Because here is this is the case where the data set is complete for mammals, so the only new data that need to be generated are for non-mammalian species. So here the case 1 is again when the data set is complete for mammals. So the T-mediated adversity is sufficiently investigated. We are able to conclude based on the lack of effects on all the available studies and parameters, and we need to generate further data on non-mammalian species to follow up as recommended in the guidance. And then currently in the guidance we recommend the OECD 231, so the AMA. Now in this specific case, so in the absence of effects on the mammalian dataset, we think that the XETA is a suitable test and maybe considered to follow up.
Okay, thank you for this clarification. Then we have a comment from Laurent Lagadic on the slide 7. And this comment is the following. So this slide is based upon the outcome of the OECD validation exercise, where a limited number of reference chemicals were tested. In a joint research project between Watchfrog and ECPA, a more recent result on additional chemicals indicate that modulators of TH clearance, including UDPG modulators, are not detected by the XETA, which is similar to the AMA. So far, phenobarbital rather appears as an exception for which more investigation are needed to explain why it elicits a response in the XETA. The results of this project were recently made available to EFSA. Would this additional information be taken into account during the revision of Annex A, Maria?
So thank you, Laurent. Yes, I acknowledge that we received the information from CropLife. And indeed, as we said, all the available information and feedback received will be taken into consideration. In this specific case, I must say, first, I mean, we just received a table, and as you may know, we don't consider data as such if we don't see the raw data. So we need really to look at the raw data to be able to use the results. With regard to the liver enzyme induction, I think this is also quite a special case, because of course when we talk about the liver enzyme induction and the metabolisms, of course there may be difference in the metabolism when we test different species, because for example, if we think at the human relevance is always discussed with regard to the liver enzyme induction, because we know that there may be difference. So it can be that the difference are really due to the difference in the metabolism that may occur when we test different species. So and I think indeed, as you say, this may be the case also when with a test according to the amphibian metamorphosis assay. It's not really about the XETA versus the amphibian versus the AMA, because in the case of phenobarbital, then also the AMA would be negative, so it has really to see why the XETA is negative. Is the AMA expected to give different results or still the same result, and why it may be different? But of course, as I said, we already had a look. We will have again a closer look and see if we can really give some more explanation, or we can include some additional information in the annex.
Okay, thank you for this clarification. So there was some additional comment on the same slide regarding the detection of TH synthesis. The question remains open that the inhibition of TPO and NIS is not consistently detected. More chemicals need to be tested to confirm the assumption that TH synthesis is not detected by the XETA. So that's not really a question, but rather a comment.
Yes. Yeah, no, go ahead. I can just say that, I mean, as we said, that of course the annex, the recommendation are based on what is currently available. Of course, when additional information becomes available, we are happy to further modify the strategy.
Okay, thank you. So we have another question. For you, Sander, when T-mediated mode of action is excluded, why proceed with testing? Can you not just conclude that it is not T-mediated endocrine disruptors?
If we talk about the case 2, then actually there is some evidence for T-mediated adversity, because it's an ED for human health. But if we talk about the case 1 in general, then no, at the moment we know that having no T-mediated adversity or activity in mammals is probably predictive of the outcome in other species, but still we need to investigate with the models that we have, because there are still differences between the route of exposure, for example, and in how they can react. So at the moment, based on the data that we have, we recommend that even if you have no T-mediated adversity in a mammalian data package, we still need to follow up with the non-target organisms, and the only models that we have available that are validated are the XETA and the AMA and the LACTA.
Okay, thank you for this clarification. So now we have a question from George Gonzalez, who is asking how reliable are the results of XETA in testing mixture of chemicals, and asking if there is data on this regard. So it's for you, Sander, maybe if you.
Upfront, I do not know how much information is available on the testing of mixtures in the XETA. The way that we use it, of course, in our biocidal active substances assessment is usually have a purified substance or at least a known substance of known composition in most cases, not all cases. So you can really focus on the substance that you are currently investigating. For our dossiers, we usually don't have any assessment like this on mixtures. And also when we consider the products, because of course for biocides we also need to consider the products as usually a sum of all the individual components that have been tested individually. And I think in the future, when people will submit XETA data, that it will be the case, that it will be an assessment of individual components of a product. That would be the base of the data. I don't think people will start testing products in the XETA as well. But for the mixtures itself, I do not know how much data is available for the XETA, but I don't think for our assessment it's the most relevant type of data.
Okay, thank you, Sander. For you, Maria, we have a question asking if the next step after a positive XETA can be an AMA to check for adversity. It is sufficient to concentrate on adversity endpoint then and leave out the activity endpoint.
So basically, if the XETA is positive according to the flow chart, so the figure 1 and the testing strategy of assessment strategy of the ED guidance, it is first recommended to postulate a mode of action, and then when a mode of action is postulated, basically if for non-mammalian species the XETA is the only available test, of course what you miss is information on adversity. And when it comes to adversity, the test that is recommended is the LACTA, because still we have to consider that an amphibian metamorphosis assay is a screening assay, meaning it is a test giving information more on endocrine activity rather than adversity. So what we recommend in the guidance is to follow up with a LACTA rather than an AMA. Then, of course, it depends on the entire data set, because if we just consider, okay, we have only the XETA, then of course we have to follow up with the LACTA as recommended. But I would like to stress that in many cases the decision or the conclusion on the outcome of an assessment is really based on the weight of evidence, where we consider all the data set together.
Okay. Thank you. Then we have another question from Thomas for you, Sander. I'm still not sure on the regulatory use of these XETA tests other than a pretest for AMA for REACH. Can please provide further information on the regulatory use?
First of all, we're here talking only about an annex to the guidance that is about pesticides and biocides, so we're not talking about REACH. So I cannot comment on how it will be incorporated into REACH. I do know that at the moment they are revisiting the REACH data requirements, so it might be that the XETA will take a similar position as we're now proposing for the data requirements for the assessment for the biocides and pesticides. But other than that, I can't really comment on the position of the XETA on the REACH.
Okay, thank you. For you, Maria, does a positive XETA need to be followed by a level 4 study, for example, a LACTA, or can a negative AMA overrule the XETA?
Well, in this case, I think it depends very much on the situation. I think basically, if we are in this situation, so that the XETA is positive and the AMA is negative, we need really to understand, to look at the case and understand why we have a different outcome. So it is a false positive, the XETA. Why? Because if there is no explanation on the reason why maybe we have a different answer between the two, I mean, as such, it's difficult to say, okay, the AMA can overrule the XETA and we don't care anymore. Also, as I said, it has to be seen in the context. Physical properties of the substance, mammalian data set, and really see whether we may explain the different outcome in the two tests. Yeah, because of course, I'm not saying that in any case, if the XETA is positive and the AMA negative, we need to follow up, but we need also to really look at the case and try to find an explanation why we have this different response, which is not expected. Because of course the AMA should cover all the potential thyroid and mode of action. So it's really not expected to have a different outcome.
Okay, thank you. So the next question for you, Sander. Would the XETA be sufficient in case no activity is observed in mammalian data package, but adversity at low dose, at dose below MTD is identified?
I think in this case, it's like Maria explained in the scenarios that you need to look at all the data together, even in case you don't reach the level of adversity, but you have indications of something happening in your model. That might be sufficient reason to stay away from the XETA and actually consider the AMA. I think a similar situation would be that even though you might not see adversity in your mammalian data package, you have some indications from QSAR or in vitro models that would indicate that still something is happening at the level, which in that case might still also allow you to consider in the AMA instead of the XETA. So it really depends on the situation. As I said, the decision scheme is a bit simplified, but you really need to look at everything together. But there can really be situations where maybe you haven't reached a level of adversity, but you have clear indications that something is happening. And then if you cannot exclude effects on thyroid production, you should not use the XETA in our opinion, and you should go for the AMA. I don't know whether Maria wants to add something on this, but.
No, I think I agree with what you have said, Sander.
Okay. So we have now a question for you, Maria. So when there are no indication from the mammalian data set that there is any T activity, and supporting studies such as bird reprod and ELS also indicate that there is no T activity, must we perform the AMA when T3 and T4 were not measured in the mammalian dataset, or can we use the XETA?
Well, this is an interesting question, I think. Basically, for the conclusion of mammals, of course, we are always involved in the discussion as ecotoxicologists, but basically, if first, I've never seen a case where thyroid hormones have not been measured, at least in one of the available studies. But having said this, if the mammalian toxicologist, based on the data set they have, and I doubt that if the thyroid hormone are not measured at all, what could be their conclusion? But nevertheless, if the mammalian toxicologists conclude that for them the ID criteria are not met because, for example, I don't know, thyroid histopathology was not affected in any of the available study, then for us their data set is complete and clean, and then we can follow up with XETA. As I said, I doubt that if the thyroid hormones are not measured, that they can conclude that the dataset is sufficiently investigated, because this is our starting point. Keep in mind that for the starting point is that the dataset for mammals has to be completely sufficiently investigated. And I doubt that if thyroid hormones are not measured, they may conclude that the dataset is complete, because I remember you that a change in thyroid hormone, even in absence of a change in thyroid histopathology, may be considered as a trigger for a DNT. So I don't know, but I'm just saying that in any case, we trust their conclusion. So if they conclude that for them the substance is not an ED, then we can follow up with XETA.
Okay, thank you, Maria. We have another question on regards to mixtures and more particularly plant extract that can be seen as mixture, for example, for thunder. So with regard to mixture, it is worth to point out that plant extract used as positive substance are a mixture of several substances. Would XETA be useful for such a mixture, or indicator active substance should be identified and then used for testing?
I think in general you'd have difficulty identifying all the individual components in any complex extract like a plant mixture. If it's defined as a substance as that, I think in principle it could be tested in the XETA, and it could be because this is your substance that is being registered. So I think in practice you could probably do that and use that result. But I'm unaware of any experience in this with the XETA. But since this is your defined substance, the thing that you want to register, I think in principle you could use the XETA if the data package would allow you to do so.
Okay, thank you, Sander. For you, Maria, so the AMA can be upgraded by prolonging the test and adding an endpoint for adversity. Would this be acceptable as a follow-up to a positive or equivocal XETA?
I must say that I have a little bit of difficulties when we talk about a modified test. Because when we modify a test, then again, we don't have experience, we don't have a validation, because the validation done when the test was published does not cover this modification. Still, we are talking about a level 3 study. So if we want to discuss about the statistical power of a modified AMA is the same as an AMA. So for the adversity, the issues are still there. And then we should also better define what is an equivocal outcome, because we may have a different interpretation of equivocal outcome. If we are really talking about a borderline case, then I don't know, because still, as I said, for me the issue is in accepting a modified AMA without further information on the performance of the test. We are not keen as regulator in accepting modification of studies without further information on the performance of the modified test. And Sander, I think if you want to add on this point.
No, I agree that the whole idea, of course, behind the validation and having a test guidelines is that you have a really standardized way of performing your test. And of course, having a slightly different way of doing something can be very informative, but it will be very difficult to start accommodating all the slight changes that you can think of in a test. So that's the whole point about having the test guideline. So it's difficult to comment on that
okay.
In general.
Okay, so maybe we can have a last question. So could the purpose of case 2 be explained again, Sander? If I understand well, it is in the case of the ED criteria being met for mammals, and therefore does it not contradict the ED guidance that states that no further testing with non-mammals is needed if the ED criteria is met for mammals?
Yeah, thank you. No, the person who asked the question, maybe I did not explain it correctly, but the point is in case 2 is that you have effects on thyroid in mammals, but which allows you to conclude it's an ED for humans, but the effects that you see are not considered to be population relevant. So even though you see an effect in an individual animal, if the effects are not considered population relevant, you do not meet the ED criteria for non-target organisms. And therefore, so your conclusion for mammals would be not an ED, and therefore you need to investigate further. So you need to investigate further with non-mammalian species. And then therefore we point to the few models that we have that are non-mammalian, which are the XETA and the AMA and the LACTA. So the point is for case 2 is that even though you see activity for thyroid and you have a mode of action, the effect itself is not considered population relevant for mammals, and therefore you need to investigate further. I hope it clarifies it better than the previous case.
Okay. Thank you very much. So we are now getting at the end of the webinar. Maria, do you want to conclude?
Yes, thank you. Let me put again the presentation. So thank you very much. So I think then the webinar has come to an end. I would like to personally thank all the participants for the interest showed in the topic. A special thank goes to my colleagues who supported us in the preparation of the webinar. So I would like to thank Francesco Amoretti, Anna Campanini, Carla Dallaglio, and Alberto Goldoni. If you have additional questions, please do not hesitate to send them to the EFSA functional mailbox that you see in the screen. The recording of today will be available on the EFSA website in a few days. And I would also like to ask you to take a few minutes to fill out the evaluation form that will be sent to you shortly after the webinar. It will take really a few minutes to fill that in, because your feedback is quite important to then improve our webinar. And if you want to keep in touch with EFSA, please use you can use our Twitter and LinkedIn accounts. Thank you very much. Bye all. I give again the floor to Sander.
So also for my behalf, thank you very much. As Maria mentioned, send all your comments to the EFSA mailbox, and then EFSA will, together with ECHA, try to incorporate your response in the revision. And I thank you so much for all your active participation and your questions. And we'll publish the annex soon, hopefully.
Odd, do you want to say goodbye?
Well, bye-bye. Thank you for your participation, yes, and thank you for your interest in the webinar.
Bye all. Bye-bye.
Bye-bye.